In this video, we will discuss further about the lipid-rich cancer-associated fibroblast differentiation. So, what are the genes that cause this kind of tumor microenvironment that has a lipid-rich cancer-associated fibroblast? And based on the analysis, what they have found out is that it's the BMP that drives this lipid-rich environment around the cancer cells, which has SETD2 knockdown. So how they come up to this point, we will understand these things using the results that are presented in this figure. So from the RNA-seq data and from the previous literatures, they have found out is that it's the BMP signaling pathways that dominates predominantly in these clusters that they have identified in the cancer-associated fibroblast and also the different kind of uh, pancreatic tumors clusters. And what they have find out is that BMP2 signaling helps in the BMP2 signaling helps in the interactions of this ductal 3 and ductal 3 tumor cells with this C2 cancer-associated fibroblast. And when they check the interactions of this C2 and ductal 3, what they have find out is that there is a high interactions of this ligand and ligand on the BMP2 to the receptors on the cancer-associated fibroblast. And here also we can see that ductal 3 has a high interactions with this C2 fibroblast in this case. And from this heat map, they are able to identify it is that ductal 3 cells are predominantly high in this BMP2. So what could be happening is that the BMP2 that are released by the ductal 3 is causing paracrine interactions with the cancer-associated fibroblast, and that in turn results in the high lipid-rich epigenetic changes in this cluster 2 cancer-associated fibroblast. And along with this BMP2, they also find out is that the BMP4 is also high in this cancer-associated fibroblast. This could be the result of the paracrine interactions of this BMP2 from the ductal 3 populations with the cancer-associated fibroblast in the cluster 2 populations. And in the literature, it is known that BMP2 and BMP4 are responsible for high lipid-rich tumor microenvironments formations. And in order to further validate this point, what they did is that they use the conditioned medium from the control and from the SETD2 wild-type pancreatic stellate cells and SETD2 knockdown pancreatic stellate cells and grow the not stellate cells, the tumor cells. So they took the conditioned medium from the control, that is the normal pancreatic cells and SETD2 knockdown pancreatic cells and SETD2 knockdown SETD2 wild-type pancreatic cells and SETD2 knockdown pancreatic cells, and then they use those conditioned medium to culture pancreatic stellate cells. And what they find out in the case where SETD2 knockdown culture medium is used, they see the more lipid-rich environment formations compared to the wild-type and control. And these things are quantified on the right hand side, where we can see that in the case of the conditioned medium where from the SETD2 knockdown is used, they see that high signals for this body IP, or we can say the high signals for the lipid-rich environment. And when they check the other factors that they have identified in the previous experiments, that is PLN1, PLN2, which is the markers for the lipid-rich environment, they see that it is high in the case of knockdown cells, and also on the other markers, that is PPARG is also high, and the clusters markers, that is ABCA8A and ABCA8B, which is the markers for the C2 cluster, is also seems to be upregulated in those SETD2 knockdown culture medium pancreatic stellate cells, indicating that in the SETD2 knockdown pancreatic cancer cells release this BMP2 or other genes also that will cause paracrine interactions with the cancer-associated fibroblast, and that might result in this BMP2 upregulations, and also both of them, and thereafter both of them might cause this cancer-associated fibroblast, or no, not both of them might cause this lipid-rich cancer-associated fibroblast formation. And in order to further validate this point that whether BMP2 is taking place or doing the same role or not, what they use is they use neutralizing antibody against the BMP2 and then check the lipid-rich marker, that is body IPY. And what they have find out in the control case, they see that increase in this body IP signals, but when they use the neutralizing antibody against BMP2, they do not see increase in this body IP signals. And same things and similar things they have. So this indicates that this BMP2 plays a role towards this generating lipid-rich environment. And when they check this lipid markers, that is PLAN2 expressions, they find out that in the case of knockdown, in the case of control, they see that high increase in this PLAN2 expressions, but when they use the SETD2 knockdown cells using alpha BMP2, they do not see too much decrease, but there is a decrease in the PLN2 expressions that can be seen here. A decrease is statistically significant, but when we compare between and yeah, the decrease between the amount of PLN2 expressions when SETD2 wild type or when the BMP2 and neutralizing antibody is not used is high, and there is a decrease in these signals when they use alpha or neutralizing antibody against BMP2. And same things they have observed for these PPARG expressions, that is decrease is statistically significant when they use neutralizing BMP2 antibodies, indicating that BMP2 plays a role towards this lipid-rich environment or lipid-rich tumor microenvironment generations. And after that, what they did is they silenced this BMP2 in the pancreatic stellate cells with SETD2 wild type and SETD2 knockdown. And what they find out is that decrease in the signals when they silence BMP2 in SETD2 knockdown is higher compared to the case where pancreatic stellate with SETD2 wild type is used. This further validate their point that BMP2 plays a role towards these lipid-rich cancer fibroblast formations. And same things they have observed in the tumors, where we can see that when in the case of control and in the case of SETD2 wild type, control as silencing RNA, and in the case of SETD2 wild type, there is no much change in the pancreatic weight formations, whereas when they use SETD2 knockdown with the silencing BMP2, they could see an increase in the pancreatic weight formations. And these things are quantified on the right hand side, where we can see that when they use SETD2 wild type, there is not much increase in the body weight, whereas when they use SETD2 knockdown cells with the silencing BMP2, they see an increase in the body weight formations. And similar things they have shown in the H&E staining, where we can see that when they use silencing BMP2, they, compared to the control case, there is not much increase in the weight or lesion tumor affected area, whereas in the case of this SETD2 knockdown cells, they see a decrease in the area. They see increase in the lesion area when they use BMP2 knockdown, and this decrease in the lesion area, yeah, because in the control case the area is high, whereas in the silencing RNAs the area is low. That can be seen quantified on the right hand side in the lesion area graph, where we can see that when they use SETD2 wild type, there is not much increase in the lesion area, neither decrease. Whereas when they use SETD2 knockdown, they see that there is a when they use SETD2 knockdown and silencing BMP2 RNAs, they see a decrease in the lesion area compared to the control case. And once they validate that the BMP2 also has an effect on the tumor weight or uh, lesion area, they check the cells effect. That is, they check the pancreatic cancer cells with SETD2 wild type and SETD2 knockdown using the silencing RNA and see whether they see an increase in this C2 cluster or decrease in the C2 cluster. In the previous experiments, they identified the markers for the C2 cluster is FAP and ABCA8A. So this is the markers they use to validate this, to check whether C2 clusters is increase or decrease, or there is a effect of BMP2 or not. And the SE2 wild type, I do not see any significant increase or decrease in any significant impact of this silencing BMP2. Whereas in the case of SE2 knockdown, they can see a significant decrease in the this C2 cluster when they use the silencing RNAs against BMP2. And these things are also quantified on the right hand side, where we can see that in the case of SG2 wild type, there is not much significant difference between the control and BMP2 knockdown cells, whereas there is a statistically significant difference between the control and BMP2 knockdown cells in SG2 knockdown pancreatic cancer cells, indicating that the C2 clusters get affected by this BMP2, which might be released by the pancreatic cells and cause paracrine reactions, paracrine impact of paracrine reactions that will result in the epigenetic changes in a way that it will cause upregulation of the BMP4, and both of them is in the literatures as known to cause lipid-rich environment. So this is how they come to a point that BMP2 derives lipid-rich cancer-associated fibroblast differentiation. If you need to understand more about these experiments in detail and how they come up to these conclusions using and wants to look some supplemental figures, you can go to the reference that has been provided in the description. That's all for this video. Thank you for your time.